really like starting talks like this with just a little bit of information about who I am and how I got here. I think it's really interesting to see the different paths that researchers have taken. So I did my bachelor's degree in mathematics at a place called Pomona College, not too far away from here. At that point in time, I thought I was going to go get a PhD in pure mathematics to study low-dimensional topology. Had a freak out at the last minute and was like, no, that's not what I want to do. And so I ended up going and working for a company called Epic Systems that makes electronic medical records. It was sort of my first view into how you can use technology for things in health and medicine. That then really led me to wanting to study computer science. So I spent a couple years at the University of Wisconsin learning about computer science and learning a little bit about biology, which then led me to do my PhD in computer science at Brown. And then since Brown, I've spent the last 10 years at this place called Carleton College. And I imagine a lot of you are like, what is this place? So it's a pretty unique kind of institution that's unique to the United States. So just to give you a little context, here is where we are right now. Carleton is way up here in Minnesota. And it's a different kind of place in that we have about 2,000 students in total. They are all undergraduate students. And we have a focus on sort of a liberal arts education. And interestingly, for us, computer science is really popular. It's been about 17% of the student body. And then something like 70 to 80% of our students go on to graduate school eventually. So these are really great undergrads. So everything you're going to see today is work that's been done entirely with undergraduate students. And I like introducing this just to let people know that there's a variety of careers in academia. So if you're curious about hearing about alternatives, I'm happy to talk to anyone. All right, so now to really dive into what we're going to talk about today. So we heard some things yesterday about tumor evolution. We're going to continue talking about tumor evolution today. I'm going to spend some time talking about a couple of brand new projects. This is the first time I've talked about this stuff before. So I'm pretty excited to show you some new things. So let's start with a little bit of background. So cancer is the result of somatic mutations. These are the mutations that occur during the lifetime of an individual. And they lead to uncontrolled cell growth and eventually the growth of a tumor. And so the types of mutations that we see in cancer can be as simple as single nucleotide variants or a single base pair has changed, but oftentimes we see other larger changes as well. So copy number aberrations, structural variants where larger segments of DNA have been impacted. But in cancer, anything goes. You oftentimes also see sort of larger changes, so complex structural variants where there are many overlapping types of these aberrations, or even whole chromosome or whole genome events. But for this talk, for sort of simplicity's sake, we're really sort of just going to be thinking about some of these simpler types of aberrations, and we're just going to think about mutations in sort of a singular category, rather than necessarily thinking about specifically which types. So as we heard from Palash yesterday, tumors are the result of an evolutionary process. So what this means is at some point you had a set of healthy cells and some founding cell acquired a first initial mutation. It gave it selective advantage. And then as time went on, different descendants of that cell acquired new mutations that also gave it a selective advantage, which means that at any point in time you're looking at a tumor, it's going to be a heterogeneous collection of cells containing different sets of somatic mutations. And so if we were able to sort of look back at time, this process is sort of the result of something that was described really nicely in 1976 by Noel as the clonal theory of cancer. And it's this idea that different sets of cells with the same somatic mutations might eventually sweep through, different populations might disappear over time, and we have these sort of evolutionary dynamics happening throughout the history of a tumor evolving. And so, unfortunately though, we don't actually have the ability to be able to observe this. The information that we get is a simple glance or a single time point or several time points when we're able to sequence a patient. And it's from there that we want to be able to infer this thing called a clonal tree that describes the evolutionary history of our tumor. So in this tree, different nodes represent different clones or sets of cells with the same set of somatic mutations, and edges are representing ancestral relationships between those populations. And so we want to use algorithms to be able to infer these trees, sort of going back in the history from a single time point. Lots of reasons why we might want to do this. Again, the more we know about the cancer, the better. I like sort of thinking about this sort of concrete example of how it might impact impact treatment. So if you think about a patient that had an evolutionary history that looked like this, if we were able to design a treatment plan that was able to target mutations on these two different lineages, this patient might be less likely to relapse because we're actually targeting mutations in all the cells in this tumor. Whereas if they had a treatment plan that maybe only targeted this blue triangle, then other cells on this different lineage might be better able to survive. And so lots of reasons why we want to be able to understand and infer this type of tree from sequencing data. So what do these trees actually look like? Here are a couple of examples. So this is a clonal tree from an AL data set. And so what you can see in this case is every node is labeled with the name of the gene that we had the mutation in. In this example, we see that there's sort of an early trifurcation and continued sort of evolution along some of these branches. One of the important things to remember here is that whenever we label a node with a mutation, that mutation then is going to be inherited by all of those descendants. We oftentimes just write it when it first appeared for simplicity in these kinds of diagrams. So unless it's otherwise indicated, you assume that everything occurred higher up is inherited further down. And this is actually, I would say, a pretty simple example of what these trees might look like. They can get a lot more complicated. So here's another example from a xenograft of a colorectal patient. And so you'll notice a lot of things happening here. So the tree is a little bit bigger. But one of the things you'll see is we'll oftentimes have these nodes that are labeled by many different genes, telling us that we weren't actually able to identify the order that those mutations occurred in. And so it's sort of indistinguishable from each other. You'll also see that sometimes we'll label these trees up here with a root called the normal or sort of healthy cell, sort of indicating that this is sort of where we started before the mutations started to occur. And again, we can see like we've got 4 branches here. There's a lot of things going on in this tree. And so one of the interesting things, though, is as people have continued to do work on this, we can infer this tree, but if I run a different method on this same data, I might get a different looking but equally complicated tree in this case. And there are some similarities here, so some of the same mutations that are higher up here are also higher up over here, but there's a lot of different things going on, and it can make it challenging to sort of really try to figure out, well, what are the differences here? What parts of this should I believe more than others? And so this has really been a really active area of research. I continually need to update this slide. This is not everything, but this is just a sampling of methods that have been designed in recent years to be able to infer these types of clonal trees from sequencing data. And so oftentimes they'll use different assumptions, they'll work on different types of data. And so really, I like showing this to convince you that there's a lot of work happening here. It's a really interesting area, and this is very much, I would say, not a solved problem. And so the work that I want to talk about today and sort of the way that I've been thinking about this isn't so much of how do I design the next method that's going to show up on this slide, but how do we work on things that are going to make the next methods better? How can we use some of this work that's come before rather than just sort of moving on to the next thing? And so the place that I'm thinking about this is how many of these methods convince people that what they're doing is good. And so many of these methods need to do benchmarking on simulated data. And so there's this idea that you're going to say, well, we don't know the ground truth on the real data that we're looking at, so we're going to create simulated data where we know what the tree looks like. I'm going to run a bunch of different algorithms, including my algorithm on that. And then I want to decide, well, which of these algorithms actually was the closest to the ground truth or gave me the best answer. There's an interesting paper that came out just in 2024 from Rachel Karchin's lab that was sort of looking at and trying to do an assessment of how people do these kinds of simulation studies. Interesting findings in there that many of these methods, groups are creating their own simulated data rather than using sort of outside sources. And so this is something I was sort of starting to really think about of like, well, how much do we really think about what goes into these simulations? What data are we looking at? Whereas oftentimes it's sort of just been like a step that you need to do on the path to get your next method out there. So the second sort of piece of this is it's not just creating the simulated data, but it's also thinking about, well, what's closer to the ground truth actually mean? And so oftentimes people use lots of different measures or distance metrics that have been designed to compare these kinds of clonal trees to determine what they mean by closer to the ground truth. And so the stuff that I want to talk to you about today is really sort of thinking about this part of the pipeline of designing a new method. How do we sort of think about simulating our data? And then how do we actually think about how we're assessing what we're getting and how good it actually is? So I'm going to talk about two projects that really sort of relate to these two pieces. So this first project, this was done by two great undergrads, Kendra, who just graduated, and Bryce, who's going to be applying to grad school in the fall. And so what we wanted to do is we really wanted to think about and look at these entire spaces of clonal trees to get a better sense of what does this space look like to help us to understand how we should be creating simulated data. And so sort of this main idea underlying this is that if we have, sort of here's a set of trees, let's say there's only 5 trees in this world, and it so happens that one of them is a green tree, we really want our samples from this data set to have effectively 20% of our samples be green trees. In reality, the space of trees that we're looking at is much, much larger than this. And so it's trying to sort of figure out what are the features that we care about and how do those actually look, or are they distributed throughout the space of trees? So our goal was to really figure out how to enumerate different kinds of clonal trees under different assumptions and being able to sort of analyze so that we can better characterize what they look like. So in order to do this, we needed to start by thinking about a different models of evolution. So we heard about a couple of these yesterday. So we started with this thing called the infinite sites assumption, also the two-state perfect phylogeny. And then the idea here is that mutations are only ever sort of gained once. Once a mutation is gained, it's never lost, so it's effectively thinking about evolution without homoplasia. This is effectively sort of the simplest assumption that we can make. And so this assumption was made in a lot of these early methods for inferring tumor evolution. And it kind of made sense of the genome is really large. Mutations are relatively rare. And so this idea that like a mutation is only ever going to happen in one place once seemed relatively reasonable, especially given the timescales that we're dealing with cancer are much shorter timescales. So this is the first evolutionary model we're going to consider, and then the second evolutionary model we're going to consider is this K-Dolo model. And so it makes a slight relaxation on here that we're now allowing mutations to be lost. And in particular, K is telling us exactly how many times the mutation can be lost. Again, this is another model that makes sense in the realm of thinking about cancer. Oftentimes there are lots of copy number aberrations, including deletions. And so this is oftentimes sort of the simplest place to people start for thinking about designing these new inference methods. So within each of these models, I'm going to think about two particular subclasses of trees. So I'm going to call this first set single label ISA trees. And really the idea here is that I'm just thinking about every clone in my tree contains exactly one mutation. So what this means is I've been able to very specifically order every single mutation that I'm considering. So in all of these sort of subclasses, I'm going to think about like this is a tree that contained 11 mutation gains. And so I'm always going to think about up to a certain number of mutations that I can consider. So single label ISA trees, they're the simplest set of trees. The next sort of generalization on that is still an infinite sites tree, but we're now allowing multi-label. And so we're representing this again as I have two integers just representing different mutations that can appear on the same node, basically just telling us that we were unable to order those particular sets of mutations. So these are two sets of ISA trees we're going to consider. I'm also going to consider two sets of K-Dolo trees. And again, we're getting as simple as we can for sort of a starting place. So the first set we're going to consider is what we call restricted one-Dolo trees. So the one meaning we're only allowing one mutation to be lost. Restricted saying that we're saying exactly one mutation. So in this case, I'm going to represent a loss by using a negative number. So here mutation 5 was gained at the root, and then it was immediately lost here. I can still have other gains, but I can only have exactly one mutation that's been lost. And then we can relax that by considering just the set of one-Dolo trees, which now allows us to have more than one mutation that's been lost. So an example in this tree, we've seen mutation 2 and mutation 1 were both lost, but they were lost once, which is why they're under the subset of one-Dolo trees. So I've got 4 sets of trees that we're interested in. And so first we wanted to figure out how do we actually enumerate the entire spaces of each of these trees. And so the single label ISA trees are the place to start. And there's actually this very nice thing that you can do using a thing called a proofer sequence, which allows you to enumerate all unrooted trees on a certain set of number of labels, and then we just have to root them to create all of the ISA trees. So the idea here is, let's say I'm trying to create a tree that contains 5 mutations. So I start with 5 nodes. A proofer sequence is going to be a sequence of 2 fewer values. So in this case, I've got 3. and it's going to contain integers that go from 1 to m, so in this case from 1 to 5. So I have a prefer sequence of 424 here, but any sequence of 3 numbers from 1 to 5 has a one-to-one relationship with one of these trees. So I could have taken any sequence of such 3 numbers. And so we're going to build this tree by walking through this sequence. And so 4 is the first value that I find, so I'm going to connect node 4 to something. And the way I'm going to do this is I'm always going to connect it to the smallest value in my tree that I haven't connected something to yet. So in this case, I'm going to draw an edge between this and 1. So 1 has now been connected, so I can never connect to that again. So my next value is 2, so I want to connect it to the smallest thing I haven't connected to yet. Well, I can't do 1 because I did that before. I don't want to connect it to itself, so it's just going to get connected to 3. And then my last value, 4, again, I want to connect it to the smallest thing I haven't connected to yet. 1 I already did. 2, it has an edge there, but it wasn't connected to, so we're allowed to connect it to that. So we add an edge there. And then this means that I have effectively 2 nodes left over that I haven't connected anything to, which in this case is 5 and 4, and so I add an edge there. And so it's guaranteed that this process is going to give you an unrooted tree, and so I can create all of my rooted trees just by rooting it in every single one of these nodes. So this is how effectively we're able to enumerate all of these single label ISA trees if we're given a number of mutations that we're interested in. This is really going to form the basis for algorithms for enumerating all of these other sets of trees. So for the multi-label ISA trees, you just start by enumerating single label trees that have... What button did I hit? Maybe the stop button on the bottom. I totally lost everything. Aha! Oh, technology. All right, so we start by enumerating all of these single label trees, and then it's pretty easy to enumerate partitions of m mutations into n sets, and so we're just able to apply those partitions to there to get all of the multi-label ISA trees. The restricted one-DOLA trees, everything is frozen.
Just click on it first and then move it.
Yeah, it's not. All right, so for the restricted one-DOLA trees, once again, we're going to start with all of our single label ISA trees. And this is one where I think it's really helpful to see a picture to sort of think through what we're doing. So what happens is we're going to take every single ISA tree, and we're going to think about how do we insert all single mutations into this tree. And so we're going to do this by we need to go through and we need to consider every single node in our tree as an insertion point. So if we start by thinking about if our insertion point is going to be right underneath 1, well, we then have to consider all ways to insert something that considers all of the existing children here. So we need to think about the power set of all of these children. So if we think about, so one of the first elements of our power set is the empty set. So I insert this mutation loss and it has no children. So all of the other existing children continue off the original node. I could have the set that's just the child 2, gets inserted this deletion so that that's a child of our inserted node. Similarly, I could have the other singleton as the child here, or I could have the entire set of children as the children of that node that we've inserted. So in this case, because we were inserting right underneath this root node, there was only one mutation that could have been deleted. If we'd had a higher or more mutations above it, we would have had to think about every mutation along that path that is a possible one that could have been deleted at that point. So then similarly, if we're thinking about our insertion point is underneath mutation 2, we can simply just add our deletion there. We also then have to consider the case that that mutation was deleted. And then our third insertion point was under mutation 3. And so we again sort of can have deletion of both mutations that exist on that path there. So this allows us to be able to enumerate all of the restricted one-DOLA trees. So then to be able to enumerate the entire set of one-DOLA trees is again going to build off of all of these steps. So we start with all single label ISA trees, and then we're going to use this same approach in kind of a clever way to sort of say I'm going to keep building my set of mutated trees. I'm going to start by adding all mutations to mutation 1, and then to that set I'm going to add all deletions to mutation 2, and then to 3, and then to 4. And you're able to get the entire set of trees in this way. So we have these 4 algorithms, and so we wanted to use them to generate some data sets to be able to analyze. You might have noticed as I was describing these that these algorithms get real slow, real quick. So we created a lot of data. I find it really interesting to think about. So the single label ISA trees is the fastest. If I had generated up to 13 mutations, I would have had 23 trillion trees, which is more than the estimated number of trees on planet Earth. A lot of trees. But the nice thing then is this is really sort of giving us a starting place to be able to go through and say, what actually are features of everything in this space? Can we actually start to analyze this to get a better idea of what we think things should look like? And so we looked at a number of different measures. I'm just going to show you a few of them. And so we thought about things that were like topological measures, so things like the height of the tree or the average branching factor. And we're able to sort of see some patterns that we might expect across these sets of trees. So if we're thinking about tree height, it makes sense that these multi-label ISA trees are going to have the shortest height because nodes or mutations are getting clustered together. And we sort of see similar growth patterns amongst these three. The one dolo trees, it makes sense again that they're going to be the tallest and their height is growing exponentially faster. But we also see some things that we didn't necessarily expect. So sort of seeing that for average branching factor, it seems like all of these sort of are converging around to a similar value in here. We also looked at label-based measures. And so not just thinking about the topology of the tree, but where the labels are. And so these are just a couple of examples of where we were thinking about, if you look at any node in your tree, how many mutations are actually being inherited at that particular position, and how does that vary? So this was great. Again, it gave us a sort of a nice place to have some data to really be able to think about what do things actually look across these entire spaces of trees. But this is a lot of work to be able to generate these trees, a lot of space to store them, and it would be really nice to be able to actually think about how do we sample from these spaces of trees now that we know some things about their features that we care about. So I'm going to step back and think about just ISA trees for a moment here. And so we started with, well, what's the simplest way that we could think of to sample random ISA trees? And there's a pretty obvious sort of iterative way of I create all my nodes, I choose a random node to be my root. Now I choose another random node and I randomly connect it to something that exists in my tree. I only had one node, so my 4 gets connected to my 3. 2 is my next random node I want to connect. I can connect it to 3 or 4. I pick one randomly, and I can build my tree up in this way. And this is a method that's been used in a lot of different papers for creating their simulated data. But if we look at what actually happens when we sample trees using this approach and we compare it to the distribution from our true set, we can see that we're actually getting trees that are much shorter than if we were looking across our entire data set in this case. And so it's sort of showing that the importance that we really need to think about how we're creating this simulated data and how much does it really reflect what it is that we want to actually be testing. So there's lots of ways that you could sample actually uniformly from this space. One cool one we ran across was this thing called Wilson's algorithm, which is initially used to be able to sample spanning trees, but it allows you, and we can see we did a random sampling of a million of these trees with 10 nodes, and we can see that in this case we're actually getting something that much better matches what we see in the actual data set. So there's still a lot of work to do here. We've just been sort of starting with the ISA trees, starting to think about how we better sample from the space of K-Dolo trees is sort of another thing for us to be thinking about. But I think it's sort of, it's a nice little project to sort of show the importance of thinking about some of these things that are not always the highlight of the projects that we're thinking about, and that they can be really important and impact sort of our downstream results. So with that, I want to shift and I want to briefly talk about another new project. And this has really been thinking about how we visualize different clonal tree distances. And so this is work that was done by Thea, who's going to be starting a PhD program this fall, and Kwok, who just finished his first year at the University of Minnesota. And we partnered with a colleague of mine, Eric, who's an expert in the field of visualization. So this project's a little bit of a departure for me. There's not so much of the algorithms involved, but really sort of thinking about how we use the output of some of these algorithms. So I want to use just a toy example of here's 2 clonal trees. They're on the same set of mutations. They're pretty different. You know, this one's pretty linear, this one's very branching. And so we could use lots of different distance measures to compare these. Parent-child distance is a really simple distance measure, and the way this works is it goes and it computes all of the pairs of parent-child relationships in each tree. So A is parental to B, B to C, C to D. Similar, we do the same thing in tree 2. And then to compute the distance, we're going to look at the symmetric difference between these sets. So what are the parent-child pairs that exist in one tree but not the other? In this case, I've got 2 over here and 2 over here, so I end up with a parent-child distance of 4 in this case. Where this gets interesting is let's add a third tree to this set. So here are 3 trees all in the same set of mutations that all have exactly the same distance from each other, but they're actually pretty different in how they're differing from each other. And just by reporting a singular number of their distance, we're losing a lot of this information about what's actually different in these cases. And so you see similar things even if you choose different, more complicated distance measures. So ancestor-descendant distance is a similar type of measure. It's just looking at longer range relationships. And in this case, we'll see that we have the same distance from tree 2 to tree 1 and tree 3 in this case. And sort of showing the importance that we're really not able to communicate the same information about exactly what these differences are, where they're coming from. And so we wanted to think about different ways to visualize this kind of information. So we've been building a tool called Vector, and sort of here's the overview of what it looks like. We've got a number of different visualizations in it. Just so you know, we're looking at data here that's from a triple-negative breast cancer patient with 2 trees inferred by 2 different methods. And so what I want to do is I just want to take a quick look at these different visualizations that we have in here and what they're telling us and allowing us to do. So most people are really familiar with the traditional node link diagrams that we've been looking at all along, but we're encoding some additional information in these. So in this case, we're looking at parent-child distance. And what we're able to do is we add encoding using colors and width of edges to sort of indicate which edges are actually contributing to that parent-child distance. So in this case, these dark edges here are telling us that, hey, there are pairs of mutations along here that are giving us unique parent-child relationships in this case. We're also using things, although it's harder to see, the size of the font and the boldness of the font. And so this allows us to at least sort of say, hey, this part of the tree are where the differences are. But on trees like this where we have mutations that are clustered together, it's still a little bit more challenging to identify, well, which mutations exactly are the ones that are contributing? And you could spend a long time staring at this and try to pull it out. But we thought it might be useful to have another type of visualization that allows us to see that information a little more clearly. So this is where we're going to use this second visualization up here, which is really just a heat map. And so in this heat map, I've got my sets of individual mutations along here. And we're using sort of colored boxes that indicate to us the distinct relationship in the 2 different trees, using 2 different colors to tell us which tree that it came from. So in this case, if you mouse over and highlight F4, you can see, hey, this is actually a child of, or it's a unique relationship as a child to all of these different parents in both of these different trees. Although you also want to think about what's showing up on the orthogonal axis on here is telling you the other relationship for here. And so you can quickly, easily look at this and sort of scan and see a couple things that are going to jump out to you as this mutation's involved in a lot of these differences. They're more likely to jump out if they're near the root of the tree or down near the leaves of the tree. We're also using other encoding to tell us when there's mutations that are unique to one tree or the other, which is oftentimes something that happens. Different methods will use different subsets of data. And so this was a useful visualization, but we really wanted to see something that allowed us to think a little bit more about how these mutations fit in the context of the tree. And so this is really where our third visualization comes in, is this thing called a tripartite graph. And so here we have all of our mutations along the middle. I have these sort of circles on either side that are indicating the same mutation. And so now we're using edges to tell us things about these different types of relationships. So the mutations in the middle are going to be children to the mutations on the left, and their parents to the mutations on the right. So an edge here is telling us that, hey, this mutation is a child of AKA P9. And so where this can get really useful again is you can mouse over these, and it allows you to sort of see all this information at once, so that, hey, AF4 has all these unique relationships as children to these sets of mutations, but it has unique relationships to parents of these other ones over here. So again, the idea was trying to figure out how to create a way that was going to give researchers an easier way to be able to figure out exactly what was going on and exactly what was contributing to these different distance measures. So everything I showed you here was using this parent-child distance, We have different encodings for a couple different distance measures in here, including ancestor-descendant and a distinct lineage distance. The last thing that I will show you is we also, because we ideally see this as being part of benchmarking, that we have a mode that you can click on here to identify one of these trees as your ground truth, so that you can see exactly what are the changes or effectively the errors from the ground truth in one tree to the other tree. So there's still a lot of work to do on that project, but I think it's sort of an exciting, sort of interesting way to be thinking about how we might do some of this stuff better. So sort of thinking forward, again, a lot of my work is really thinking about this slide. I've done a bunch of things. I talked about a bunch of stuff related to simulating data and benchmarking today. There's a lot of interesting work in there, or thinking about things like the space of clonal trees. I'm really curious to think about how this space clusters onto different modes of evolution. And so there's been papers out there thinking about linear or branching, or sort of like Big Bang models, and thinking about how that looks over across the space of these trees. I've done a bunch of other work thinking about other sorts of ways of how we utilize these methods as a whole, so thinking about maybe how we take consensus trees across different methods that are giving us different possible inferences of tumor evolutionary trees. So again, really thinking about this work from a slightly different angle of how do we improve upon or make better use of everything that exists, rather than just what are the new things that we need to create. So with that, I would like to thank the students who did all of this work, sources of funding, and all of you for listening. Thank you.